Hello friends, welcome to my channel Sun Biology Class. Today we will discuss about Rt-Pcr. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Rt-Pcr means reverse transcription polymerase chain reaction. It is a laboratory technique. It is a laboratory technique that combines reverse transcription that combines reverse transcription and polymerase chain reaction and polymerase chain reaction PCR. Reverse transcription is a process by which DNA is synthesized on RNA template. This DNA, which is complementary to the RNA template, is known as complementary DNA or cDNA. On the other hand, polymerase chain reaction (PCR) is another technique by which specific DNA sequence can be amplified. By Rt-Pcr, the target RNA sequence is first reverse transcribed into cDNA by the enzyme reverse transcriptase, and then the cDNA is amplified by polymerase chain reaction to yield very large number of cDNA copies. Very large number of cDNA copies. Rt-Pcr thus combines reverse transcription and polymerase chain reaction. Rt-Pcr is commonly used to measure the amount of specific RNA in different samples. Based on whether reverse transcription and polymerase chain reaction are carried out in the same or in separate reaction tubes, Rt-Pcr is divided into two types. First one is one-step Rt-Pcr. First one is one-step Rt-Pcr in which both reverse transcription and polymerase chain reaction are carried out in the same reaction tube. This approach minimizes experimental variation and is less prone to contamination. Second one is two-step Rt-Pcr. In this technique, reverse transcription and polymerase chain reaction are performed in separate reaction tubes. This technique is more susceptible to contamination due to more frequent sample handling. First step of Rt-Pcr is reverse transcription. Reverse transcription. It is a process by which DNA is synthesized on a RNA template by the enzyme reverse transcriptase. Reverse transcriptase cannot initiate DNA synthesis de novo. It can add deoxynucleotides only to a pre-existing DNA chain. To carry out cDNA synthesis, reverse transcriptase requires primers. What is primer? Primers are short DNA. Primers are short DNA oligonucleotide. Oligonucleotide. Primers are best paired to the RNA template and provides the starting point for the addition of successive nucleotides by reverse transcriptase. The function of the primer is to provide a free 3' hydroxyl group to which nucleotides are added, thereby extending the primer in 5' to 3' direction. In reverse transcription, 4 different types of primers are used. First one is oligo-DT primer. First one is oligo-DT primer. Second one is anchored oligo-DT primer. Third one is random primer. Third one is random primer. And last one is gene specific primer. Gene specific primer. Eukaryotic mRNA is characterized by the presence of a polyetail at the 3' end. Polyetail is a sequence of RNA containing only adenylic acid residues. All such RNAs containing polyetail can be reverse transcribed into cDNA by using oligo-DT primer and anchored oligo-DT primer. What is oligo-DT primer? Oligo-DT primer is a string of deoxythiomidylic acid that pairs with the polyetail and then provides the starting point to begin the synthesis of complementary DNA by reverse transcriptase. This enzyme extends the primer by adding nucleotides to the free 3' hydroxyl group of the growing DNA chain. As a result, the new DNA strand is synthesized by reverse transcriptase in 5' to 3' direction. This newly synthesized DNA, which is complementary to the RNA template, is known as complementary DNA or cDNA. One of the disadvantages of using oligo-DT primer is that it can also bind to the internal poly sequence which may be present within the RNA. If the oligo-DT primer will bind to the internal poly sequence, then reverse transcription will result in the formation of a truncated cDNA from which a part of the sequence is missing. This problem can be overcome by using anchored oligo-DT primer. Anchored oligo-DT primer consists of a string of deoxythiomidylic acid followed by 2 additional nucleotides Vn. These 2 nucleotides form the anchor sequence. Anchor sequence. Out of two nucleotides, V can be deoxyadenylic acid, deoxyguanylic acid, or deoxycytidyllic acid. On the other hand, N can be any one of the four deoxynucleotides. The presence of the anchor sequence ensures the binding of the primer to the 5' end of the poly tail. It also prevents internal priming and ensures the reverse transcription of the entire mRNA into cDNA. Like DNA replication and transcription, reverse transcription also takes place in 5' to 3' direction. In addition to this, for reverse transcription, random primer and gene-specific primer can also be used. Random primers like hexamers are non-specific. They can bind to any RNA sequence, hence is useful for the synthesis of cDNA from a wide variety of RNA species. On the other hand, gene-specific primer contains specific sequence that ensure its binding to a particular region of RNA template. Such type of primers are useful in the reverse transcription of specific RNA into cDNA. Second step of Rt-Pcr is polymerase chain reaction by which cDNA copies are amplified and analyzed. Based on whether the cDNA sequence is analyzed at the end of the process or in real time, there are two different methods. One is endpoint Rt-Pcr. One is endpoint Rt-Pcr and the other is real-time Rt-Pcr. Real-time Rt-Pcr. Both are variations of the polymerase chain reaction. They only differ in how PCR products are analyzed. In endpoint Rt-Pcr, the amplified cDNA copies are analyzed only at the end of the process using fluorescent dye such as ethidium bromide, by gel electrophoresis. By contrast, in real-time Rt-Pcr, the amplified cDNA copies can be analyzed in real time. That means while the amplification process is still going on. This is achieved by using fluorescent probes. Such as Tachman probe, molecular beacons, and scorpion probes. Friends, to know more about conventional and real-time polymerase chain reaction, please watch my previous videos. Rt-Pcr has several applications. First one is it is used in research to measure gene expression. Rt-Pcr is also useful in the insertion of eukaryotic genes into prokaryotes. In addition, this technique is also useful in genetic disease diagnosis, cancer detection, and pathogen detection. Friends, this is all about reverse transcription polymerase chain reaction. For more updates, subscribe my YouTube channel Sam Biology Class.